In this video, we are going to be discussing proteins, and this is going to be the first of two videos. And in this one, we're going to be discussing amino acids. Those are the monomers of proteins, and peptide bonds, how those amino acids are connected to one another to make a protein, to make a chain of amino acids. And by the end of this video, what I want you to be able to do is I want you to be able to identify the functional groups of amino acids. Okay, so if you don't remember what those functional groups are, go back to the functional group video and watch that again. In fact, I encourage you to review that again, and we'll discuss why it's so important in just a few slides here. I want you to be able to categorize the R groups of amino acids. So there's several categories I want you to be able to look at a amino acid and be able to determine which category it belongs in. I want you to be able to draw the structure of a glycine amino acid. So there's 20 different amino acids. Some of them can be pretty complex. For the nature of this course, for the purposes of this course, glycine is the simplest, and I want you just to be able to draw that one. I want you to be able to draw a peptide bond that connects two glycine amino acids together. And on a polypeptide, I want you to be able to identify the carboxyl and amino terminal ends. Okay, so let's get started. Proteins are absolutely essential for life. In fact, if you were to take a look at your cells or cells of any organism, whatever is not actually made of proteins, and lots of structures within cells are made of proteins, but even the things within a cell that aren't made of proteins were probably required a protein to be made. Okay, so if we take a look at luciferase of a firefly, proteins, hemoglobin within red blood cells, proteins, keratin within these horns within your hair, proteins, proteins and hormones and antibodies and transporters and muscle fibers and spider webs and feathers and the lens of your eye, proteins, 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 proteins. Now, there are other things within your cells that are not themselves proteins. There are important structures of your cell that are carbohydrates. There are important elements of your cell that are made of lipids. But here's the thing. One of the most important types of proteins are enzymes. And enzymes are biological catalysts. Right? What that means is they speed up chemical reactions. In order to make a certain carbohydrate within your cells, you need an enzyme to catalyze the reaction, to catalyze the production of that carbohydrate. That enzyme is a protein. In order to make a proper phospholipid or another type of lipid within your cells, you need to speed up those chemical reactions, and you do that with an enzyme, a protein. So proteins really are absolutely essential for your cell's structure and your cell's function. And there is an absolute diverse array of functions for proteins. There are, as we already mentioned, enzymatic proteins. There are defensive proteins like antibodies. There are storage proteins like ovalbumin in eggs. There are transport proteins that allow the cell to get things into or out of it. There are hormonal proteins like insulin that tell cells, right, the state of the body, whether we have too much or too little glucose. There are receptor proteins. There's contractile and motor proteins of your muscle. There's structural proteins in connective tissue like collagen. Now, each one of these, in fact, probably every one of these types, we're going to go into more detail in this class. So just have an idea and a few examples. But again, we will see pretty much all of these again later on within the course. All right. Proteins are our third polymeric macromolecule. So if we go and take a look here that this table that we've created before, we talked about carbohydrates, the monomers, monosaccharides, the covalent bond are glycosidic bonds. We've talked about nucleic acids. The monomer of nucleic acids are nucleotides. The covalent bond that connects nucleotides together is a phosphodiester bond. And now we're here in proteins. And we're going to spend a lot of time with proteins. The monomer are the amino acids. We're going to discuss those in quite a bit of detail in a moment. And the covalent bond that connects amino acids are peptide bonds. So let's take a closer look at an amino acid. Every single amino acid has the same basic structure. It has what we call an alpha carbon, and connected to that alpha carbon is a carboxyl group, and because it's connected to the alpha carbon, we call it the alpha carboxyl group. We have an amino group, okay, and because it's connected to the alpha carbon, it's called the alpha amino group. We've got a hydrogen, so now we have 1, 2, 3 covalent bonds of the 4 possible for a carbon. So we now have a 4th region, and this right here is where each amino acid is different. It's called the R group. There are 20 different amino acids, and therefore there are 20 different R groups. Okay? So as you can imagine, because every single amino acid has at least one amino group and at least one carboxyl group, those 2 functional groups are going to be really important to understand. And we'll talk about them in more detail in a moment. And of course, we've talked about them in great detail in your functional group lecture. Now, let's get back to the R groups here. There are 20 different amino acids, and therefore there are 20 different R groups, but based on the chemical characteristics of those R groups, we can sort the 20 amino acids into 3 categories. Amino acids with nonpolar R groups, amino acid with polar R groups, and amino acids with charged R groups. So let's take a look at the amino acids with non-polar R groups. And I've got a question here. If we were to take a look at this, and I didn't explain anything to you, how do you know that these R groups are non-polar? And in each one of these examples, it's in yellow. The R group is in yellow, right? Because this is the alpha carboxyl group, that is the alpha amino group, and that is the alpha carbon, which means that must be the R group. Notice, some R groups can be quite complex, but take a look at glycine. Its R group is nothing more than a hydrogen. In fact, I want you to be able to, from memory, draw the structure of glycine here with its single hydrogen R group. Now, how do we know that these are nonpolar? We'll take a look. Carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. These R groups are made up of nothing more than carbons and hydrogens for the most part. And because of that, because we know the electronegativity of carbon is equal to that of hydrogen, therefore these R groups are nonpolar. Okay? They cannot interact with water. They cannot form hydrogen bonds. Let's take a look at the next category. So these are R groups, or also called side chains is another term you'll hear, and they're polar but non-ionic. So again, I asked the question, what makes these R groups polar? If I were to give you some amino acids and ask you, are the R groups of these amino acids polar, how would you be able to figure that out? Well, take a look. You're looking for very electronegative atoms connected to not so electronegative atoms in the R groups. Aha, an oxygen and a hydrogen. We know we've got some polarity there. Partial negative charge on that oxygen, partial positive charge on that hydrogen. Another hydroxyl group there. Here's a sulfhydryl group. Here's another hydroxyl group, right? So it's fairly easy to look at those R groups and say, yeah, those are polar. And finally, the third category are ones that have the ability to exist in an electrically charged state or an ionic state. We've got ones that can be negatively charged, and we've got ones that can be positively charged. And take a look at why that's the case. Look, this is a carboxyl group here. And notice it's not the alpha carboxyl group. It's a carboxyl group here in the R group. Here's another carboxyl group. This is an amino group. Right? So we're seeing carboxyl groups, we're seeing amino groups not only in the backbone of the amino acid itself, but also in its R group. And I have a tricky question here. Will the R group of aspartate, here's aspartate, here's the R group, will it always be charged? And to answer this question, we have to go back and think about carboxyl groups. And remember, let's go over here, a carboxyl group. So let's say this is our R group, and it's connected all the way at the end of that carboxyl group. And right now, as the image shows you, it is negatively charged. But what did we learn about carboxyl groups? We learned that when you put them into an aqueous solution, they reach an equilibrium. Some of them exist like this, but some of them will grab a proton from the aqueous solution and become this. Right? This is the deprotonated state. And this is the protonated state. Protonated. And notice, this is so important. The deprotonated state has a big fat negative charge, and the protonated state has no charge. That's going to become very important when we start discussing the nature of protein structure in a little bit. But what that means is the answer to this question, will the R group of aspartate always be charged? The answer is no. That sometimes, if the proton concentration in solution is high enough, sometimes those protons will bind to this, and you'll be in that non-ionic state of a carboxyl group. So again, I hope I've made this clear that one thing you should focus on is really learning about carboxyl groups and about amino groups, and go through each and practice assigning these terms: protonated versus deprotonated, charged versus uncharged, acidic form versus basic form. Be able to do that with carboxyl groups, be able to do that with amino groups. And if you can't remember how to do it, go back to the video on functional groups, and I go through in pretty good detail about that stuff. Okay, so now you have an idea of what is similar amongst all amino acids and what's different amongst the 20 different amino acids. And you should be able to take a look at an amino acid, look at its R group, identify its R group, and determine what category that R group belongs into. Is it nonpolar? Is it polar? Or is it electrically charged? So finally, let's take a look at how we connect two amino acids together. How do we make a polypeptide? How do we make a protein? So polypeptide and protein, those two terms are essentially similar, or synonymous, excuse me. So what we have here is this is the growing polypeptide. Here we have the alpha carboxyl end. This is the alpha amino end, or the N-terminal end, or the C-terminal end. What's going to happen, and this is the new amino acid coming in, what's going to happen is we're going to lose an H from the alpha amino group of the incoming amino acid. We're going to lose an OH from the carboxyl, the alpha carboxyl group of the end of the peptide chain. There's our water, so this is a dehydration reaction, and there is the peptide bond. So that peptide bond is between what was once the alpha carboxyl group and the alpha amino group of the incoming amino acid. And so no matter how long our polypeptide chain gets, we're always going to have the N-terminal end, and we're always going to have one C-terminal end. And we just keep adding to the C-terminal end, adding to the C-terminal end, adding to the C-terminal end. Okay? Make sure that you can link two glycines together via a peptide bond. Okay, so that's the end of our first part of proteins. And now I hope that you can identify functional groups of amino acids. I hope you can look at an amino acid and determine what's the alpha carbon, alpha carboxyl group, alpha amino group, and the R groups. I want you to be able to then put those R groups into those three different categories that we discussed. I want you to be able to draw the structure of a glycine amino acid and connect those two amino acids together via a peptide bond. And I want you to be able to identify the carboxy and the amino terminal ends of a polypeptide.